Thank you very much, Marcia, and thank you for the opportunity to share our work. Today, I'll share with you some data on how the injured myocardium can mobilize neutrophils from the spleen, and it induces transcriptional activation of these neutrophils in the blood before they arrive at the injured myocardium. The majority of this data is unpublished, and I'm keen to hear your views. The etiology of acute myocardial infarction is well known. Risk factors such as a sedentary lifestyle, smoking, a diet high in cholesterol, and genetic factors such as ethnicity influence the risk for myocardial infarction. But despite the number of causes, the underlying pathology is atherosclerosis, so fatty depositions in the coronary arteries, which over decades become very active. And the recruitment of immune cells results in an atherosclerotic lesion, which leads to vast remodeling of the vascular tree. A rupture or a blockage in one of these coronary arteries will result in myocardial infarction, where you get localized damage to the ischemic heart. The primary therapy for MI is percutaneous coronary intervention, or PCI, where a treating physician will remove the clot. Sometimes they don't. They'll implant a stent, they'll reperfuse the myocardium in order to salvage it. This is very effective and partly responsible for the decline in the number of deaths that are associated with myocardial infarction. But once the heart is injured, in the subsequent hours and days, it sustains secondary damage mediated by immune cells. In the immediate hours following myocardial infarction, you get a robust immune response, which results in elevated blood neutrophils, which are present as the patient arrives at the hospital. So it's very, very quick. After a heart attack, a substantial number of neutrophils will infiltrate the injured heart in the immediate hours. Here, they will activate, they will degranulate, and they will prime the inflammatory environment for the subsequent arrival of monocytes, which are well studied in the setting of heart injury and repair. Depletion of immune cells, or inhibition of their recruitment to the heart, lowers the final infarct size, but it doesn't always result in a better outcome or better function. And that's because immune cells mediate both aspects of further injury following myocardial infarction, and they also mediate repair in the subsequent days and weeks. So neutrophils are the most abundant immune cell in peripheral blood and in the injured heart in the first 24 hours following injury. They're the earliest immune cell to arrive, and their number in peripheral blood as the patient arrives at hospital is predictive of how big the injury is going to be, the extent of myocardial damage. But how neutrophils are recruited to the heart, how their gene expression profiles are regulated, and where the cells are recruited from remains largely unknown. A better understanding of these processes would allow us to target the first cells that infiltrate the injured heart in the immediate hours after injury to salvage the myocardium from inflammatory damage. So we have hypothesized that neutrophils can be mobilized from reserves such as the spleen or the bone marrow, and that the injured heart is able to signal this mobilization. We further hypothesize that analysis of neutrophil gene expression profiles will allow us to identify pathways and processes that are amenable to therapeutic intervention. So in Oxford, we benefit from a large clinical study called the Oxford Acute Myocardial Infarction Study, where patients presenting with myocardial infarction are recruited, and then they're highly characterized with respective cardiac magnetic resonance imaging and serial blood sampling. In agreement with previous studies, we find that the number of neutrophils in the blood of the patients as they arrive at the hospital correlates with the extent of myocardial injury as determined by MRI imaging. So in these graphs, you can see that the number of blood neutrophils in the patients as they come into the hospital is on the y-axis, and the extent of injury is a surrogate marker. It looks at the amount of water or edema in the heart, and then this correlates with the final infarct size. And we can quantify the size of the scar or the size of the infarct 6 months later in the same patient using late gadolinium enhancement. In both measurements, which are the same patients, more neutrophils in the blood results in a larger infarct. The rapid elevation in peripheral blood neutrophils in the immediate hours following myocardial infarction is indicative of mobilization. But where are the cells mobilized from? To investigate whether the spleen or bone marrow mobilizes neutrophils after injury, because they're reserves, I used a mouse model of myocardial infarction where we ligate the coronary artery, allow the animals to recover, and harvest tissues of interest such as the blood, spleen, bone marrow, or heart, and determine neutrophil populations using flow cytometry. We find that there's a rapid increase in peripheral blood neutrophils, a simultaneous reduction in splenic-neutrophil number, no alterations in the bone marrow and the heart at this early time point. And the rest of the talk will look at what causes this early splenic neutrophil mobilization. This is exciting for us. It's not been demonstrated before that neutrophils that reside in the spleen can be mobilized following myocardial infarction. And we think that's because traditionally research groups like ourselves, who are interested in immune cell kinetics and immune cell activation following injury, we don't look at very early time points. But we also hypothesized that the mobilized peripheral blood neutrophils would undergo alterations in gene expression before recruitment to the injured heart. This is similar to what we have previously reported in monocytes, which show prior activation or programming in patients and in mice before they undergo recruitment. To determine whether neutrophils show priming or programming, I used RNA sequencing. Here we have N-STEMI control patients and STEMI patients at the time of presentation as they come into hospital, and then we follow up the same patient 1 month later to get a matched control sample. So STEMI patients show no alteration, STEMI patients show alterations typical of a myocardial infarction on ECG, and they'll show a rise in cardiac troponin in the blood. N-STEMI patients differ because they might have a normal ECG and they may not have elevations in cardiac troponin within the blood. But upon an invasive clinical angiogram, the treating physician might find a blocked vessel and they might treat with a stent, whereas STEMI patients are always treated with a stent. Using unbiased analysis, such as this PCA plot, we find that the STEMI patients group together and that there's clear delineation between the time of presentation and then the time of follow-up. If we look more closely at the absolute number of genes that change in each group, we find that over 900 genes change in STEMI patients, which is surprising because neutrophils are regarded as cells that are transcriptionally poor. If we employ further bioinformatics to determine whether the genes that change are favoring any particular pathways, we find that there is a strong representation of inflammatory pathways with a significant enrichment for pro-inflammatory TNF-alpha and for interleukin-6. So this changes the way that we think of neutrophils because they're not just responding to the local environment in the myocardium, but they might have prior priming or programming. And this opens up the possibility of targeting these cells to alter their functions therapeutically in the setting of regeneration and repair. But how does the injured heart communicate with the spleen to induce the mobilization and the potential activation of these cells? Whilst the soluble factor from the heart could mediate the response, the concentration gradient is inverted. It's possibly very high in the heart, but respectively lower in peripheral blood and then lower in the spleen. But even if you look at the blood from the coronary sinus, so patients, we can get blood samples from the coronary vascular tree, and if you look at chemokine levels here, they're only modestly elevated. So following PCI, following treatment, there's a net increase of 0.1-fold. But the neutrophil numbers in peripheral blood are already elevated. And if you go back into the studies in the 60s, the 70s, and the 80s, and you look at neutrophil mobilization from the bone marrow, they used intravenous chemokine injections that were several orders of magnitude greater than what we detect in the coronary effluent, around 400 to 600-fold more concentrated than what you find in patients. And what we also know from patients is that you don't see a peak in blood chemokine profiles up until 24 hours after injury. And this is subsequent to the rise in neutrophil numbers following myocardial infarction. This to us suggests that other mechanisms may be responsible for the early or the immediate mobilization of neutrophils following myocardial infarction from the spleen. And we hypothesized that extracellular vesicles, which are these membrane-enclosed envelopes, they carry proteins on their surface, microRNAs within their spheres. They could mediate this effect, and I've previously shown that extracellular vesicles can mobilize monocytes from the spleen and induce their transcriptional activation. For those of you who are unfamiliar with extracellular vesicles, EVs is a broad term given to three main populations of membrane-enclosed particle. The largest of these are apoptotic bodies, which are produced by dead or dying cells. You then have microvesicles, which are derived from budding from the plasma membrane. And then you have the smallest, which are called exosomes, which are derived from the late endosome, which generates a multivesicular body. It fuses with the plasma membrane, and then through exocytosis releases exosomes. There's significant overlap between all of these populations, and it's difficult to determine the subpopulations accurately in complex biological fluids such as plasma. But you can isolate extracellular vesicles from biological fluids using ultracentrifugation, where initially you will pellet down cells and debris. Using increasing centrifugal force, you can pellet down microvesicles, and then using more force, you can pull down exosomes. The data I'll share with you today is a combined mixed population of so-called microvesicles and of exosomes, but I'll call these EVs. The isolated EVs were washed in PBS following isolation, and I'll cover the characterization as we progress through the data. There are a number of other methods you can use to isolate extracellular vesicles, and I've confirmed some of our findings using methods such as size exclusion chromatography, and we're in the process of repeating some of this analysis using immunoaffinity capture against tetraspanins, which are present on extracellular vesicles, and integrins. But in the interest of time, I won't go into the benefits of each method and the limitations. So we tested the hypothesis that extracellular vesicles mediate splenic-neutrophil mobilization following myocardial infarction. I isolated plasma EVs at the time of presentation, so as the patient comes into the hospital, and then 6 months later, in the same patients to serve as a control. So patients presenting with myocardial infarction have significantly more EVs at the time of injury versus a 6-month control measurement, but they have similar size profiles with a potential increase in the small exosome fraction. So the concentration of the plasma EVs is on the y-axis, and the size of the particle is on the x-axis. And then this is just showing the grouped data at presentation and 6 months later. Plasma extracellular vesicles of any nature require that you characterize them. One way you can do this is to look at their morphology using transmission electron microscopy. This is quite an old photo from a couple of years ago, but what you're looking for is a membrane-enclosed particle, and sometimes they can have a collapsed cup-shaped morphology. What you're also looking for in your EV preparations are the presence of tetraspanins or EV markers, such as this CD9 and CD63. So the plasma EV number is elevated after myocardial infarction, but does it correlate with the size of injury? So plasma EV number at the time of presentation correlates with how big the myocardial injury is and how big the scar is at 6 months. These are the same patients I showed you earlier. So a larger EV count equals a larger infarct. by quantification of the edema, which is the extent of injury, and then using late gadolinium enhancement, which is the size of the scar at 6 months. We also find a highly significant relationship between the number of plasma EVs at the time of presentation and the number of plasma neutrophils at presentation. Now, although this is just an association, it raises the question that EVs may mobilize neutrophils following MI. EVs are elevated in the peripheral blood following myocardial infarction, and neutrophils are also elevated, and they both correlate with the extent of cardiac injury. EVs contain bioactive molecules or substances such as microRNAs, and are these altered following myocardial infarction? To determine this, we isolated plasma extracellular vesicles again at the time of presentation, and then 6 months later, we used the reverse transcription qPCR array, and we investigated over 700 microRNAs. And what we find is that there are 12 microRNAs that are significantly elevated in the plasma EVs at the time of injury. This is a slight oversimplification of the model that we used. We also use an elastic linear regression model where we tell it the size of the infarct, the EV density, and other clinical characteristics to determine which EVs are significantly altered. There are many cells within the heart that can generate EVs, and indeed EVs which are enriched with microRNAs. But all of these microRNAs are present within endothelial cells, and then 2 of these microRNAs, so microRNA 1263P and microRNA 1265P, are endothelial cell associated or endothelial cell specific. And microRNA 126 is of particular interest to us because endothelial cells are a highly plausible source for the generation of the myocardial signal for potential immune cell recruitment. In ischemia, potentially from the spleen. They're in very close proximity to the infarcted area. They're bathed in the ischemic environment, and they may produce and release extracellular vesicles to signal immune cell recruitment. Therefore, to examine whether endothelial cells produce EVs, I used a model of human endothelial cell activation in vitro and isolated the EVs. Here I've cultured primary human endothelial cells. We stimulated them with a pro-inflammatory TNF-alpha and then isolated the EVs using ultracentrifugation. So human endothelial cells produce and release EVs under basal conditions, and if you stimulate them with a pro-inflammatory factor TNF-alpha, they significantly release more. And if we characterize these, we can show that they have the typical cup-shaped morphology, and they're positive for EV markers such as TSG101 and the tetraspanning, again, CD9. So here on the Western blot, I've used the sham media only control to account for any bovine-derived EVs in the culture media. And this is an important control because bovine EVs carry bioactive material as well, such as microRNAs. And some of those microRNAs show homology to the mouse and the human. So if you're planning on using them downstream in functional studies, you need the appropriate control. And so we use a sham media as our control. I also wanted to know whether stimulating the endothelial cells would enrich the microRNA within the extracellular vesicle fraction. So I isolated the EVs from these preparations and I looked at whether the inflammatory stimulation would increase microRNA 126. And we find enrichment in the EVs for microRNA 1263P and 5P following inflammatory stimulation, and it supports our clinical observations where we see enrichment for 126 in the peripheral blood plasma EVs in patients during myocardial infarction. So microRNAs control gene expression. And does microRNA 126 show any influence on neutrophil biology? By looking at the seed sequence of microRNAs such as 126, you can look at curated databases and determine what their predicted or their putative targets are. We do a lot of translational work, which I'll share with you shortly, but I wanted to know whether there was similarity between the microRNA 126 putative targets or predicted targets in the mouse and the human. Using in silico techniques, we can take our microRNA of interest, we can use curated databases. Here I'm using 3 different databases to increase the stringency of our analysis. And I've asked, are the targets similar amongst the species, so mouse versus human? Or do they converge on similar biological pathways? Here we employed a Fisher's exact test to assess commonality between the target list, between all protein coding genes found between the mouse and the human as targets for 126. We find an overlap of 2,760 genes, which has an intersect of about 30%. This is significant, which are present both in the mouse and the human. We further assessed commonality between the mouse and the human, microRNA 126 related messenger RNAs using gene ontology to determine whether the putative or the predicted targets in the human and the mouse cluster around similar biological functions. So using a Fisher's exact test again, we assess whether there was commonality between the mouse and the human predicted targets and the pathways that they may influence. We find that there's a significant intersect here, an overlap of about 40%, and this includes gene ontology terms for cell migration and cell motility. So microRNA 126 messenger RNA targets are similar in the mouse and the human, and there's an overlap in the potential biological functions or their target pathways. Using a similar approach, I wanted to know whether microRNA 126 could influence neutrophil biology. And so I looked at the predicted targets from the human, the mouse, and the overlap list, so the targets that appear in the mouse and the human, and then I downloaded all of the neutrophil gene ontology terms from the consortium. And then we use the Fisher's exact test again to look at the overlap, and what we find is that microRNA 126 targets are significantly overrepresented for certain neutrophil gene ontology terms for migration and chemotaxis, but not for other terms such as neutrophil clearance and neutrophil mediated killing of a fungus. So this is exciting, it fits with our hypothesis, but do microRNA 126 targets change in neutrophils following myocardial infarction? To determine this, I use the RNA sequencing data that we showed you earlier. We take the microRNA 126 putative targets from the human, the mouse, and the overlap list, and then we compare them against the number of genes that change in STEMI patients following myocardial infarction. So these are the 900 genes that are changing in the neutrophils from the patients as they come into the hospital. And we find that microRNA 126 messenger RNA targets from the human list are significantly overrepresented in the patient at the time of injury. Building on this, I wanted to know whether you would get a similar response in the mouse, so we could potentially mechanistically look at this using an animal model. I've recently contacted Clement Cochain from the University of Würzburg in Germany, and Clement and his team have performed myocardial infarction in the mouse and then used single-cell RNA sequencing of blood neutrophils. They show that there's heterogeneity in the blood neutrophil profile following myocardial infarction, and they can be grouped or clustered. Here in this single-cell RNA sequencing plot, we are showing you 6 clusters, but I wanted to know whether there was similarity again between the mouse and the human. Do the same genes change in the mouse versus the human? We have previously used this to look at monocyte responses in the human and the mouse, and it's a good way to compare species and clinical and experimental data. So using the Fisher's exact test, we find that there's similarity in genes that change between the mouse and the human following myocardial infarction, and we find enrichment for microRNA 126 messenger RNA targets in some of the neutrophil clusters. We've built on this and targeted microRNA 126 using an antagomer with Catherine Moore's group in New York, but I'm not going to share that data with you today. Instead, I want to focus on the EV response. So endothelial cell-associated microRNA 126 is significantly enriched in plasma EVs following myocardial infarction, and microRNA 126 messenger targets are significantly altered in the mouse and the human following injury. But EVs also carry protein on their surface from their parent cell, and endothelial cell integrins are well described. So I wanted to determine whether we could detect endothelial cell integrins and whether they were enriched following myocardial infarction. So endothelial cell integrins such as CD31, ICAM, VCAM1 are expressed in some endothelial cells constitutively, but also expressed following activation in response to injury and inflammatory proteins. To determine the presence of endothelial cell integrins on plasma EVs at the time of injury, I isolated plasma EV from our patients and from the mouse model of myocardial infarction, and we find enrichment for vascular cell adhesion molecule 1. But what about endothelial cells in culture? So following a similar experiment to what I showed you earlier, where we take endothelial cells in culture, we expose them to an inflammatory protein or control conditions, we can then isolate the EVs, we find enrichment for endothelial cell integrins following inflammatory stimulation, similar to what we observe in the plasma of the in vivo samples. VCAM1 is consistently enriched in EVs in all studies. This was promising and exciting because VCAM1 is regarded as a stationary molecule that is expressed in activated endothelial cells. It tethers immune cells to blood vessels, and here we're showing that it's present on plasma-liberated EVs and that it's enriched on the EV fraction during injury. One of my concerns at the time of doing these experiments was, could we possibly be isolating VCAM1 or soluble VCAM1 using ultracentrifugation, because these methods are liable to contamination. So to show that the VCAM1 was on the surface of EVs, I isolated plasma EVs, I took some magnetic particles of iron oxide, I conjugated anti-VCAM1 antibodies to the surface of these beads, used IgG as a control, and what you can do is then you can incubate your EVs with your magnetic beads conjugated to your antibodies, so similar to performing an IP, if you have experience in that, and then you can incubate them, wash them, embed them into agar, and then section them using transmission electron microscopy techniques. And what you find is that your magnetic bead is now decorated in VCAM1 positive extracellular vesicles, and your IgG is completely clear. Our PhD student, Dan, who's on the call now, is now in the process of applying this enrichment approach to our clinical cohorts. Endothelial cell-derived EVs are enriched in the blood following myocardial infarction. They show enrichment for an endothelial cell microRNA 126. They show enrichment for an endothelial cell integrin, VCAM1, but do they localize to the spleen and can they mediate neutrophil mobilization? It's difficult to determine the biodistribution of extracellular vesicles in vivo because they're very, very small. So your modal diameter is around 120 nanometers. But I've used the labeling technique that allows us to quantitatively assess tissue distribution in the mouse over time. And I've kept the species of EVs for these studies exactly the same. So we're now using mouse-derived endothelial cells to produce mouse endothelial cell-derived EVs. So similar to the human, mouse endothelial cells produce EVs under basal conditions, and they release significantly more in response to TNF-alpha, a pro-inflammatory factor. They show typical morphology, so we have this cup-shaped picture by TEM, and then if you use cryo-TEM where the EVs don't collapse because of the dehydration steps, you get a circular morphology, and they're positive for CD9 and for ALEX, which are EV markers. Again, in these studies, I've used a media sham control to account for the FBS, or the bovine-derived EVs, in our culture media. So we deplete bovine-derived EVs from the culture media by ultracentrifugation, but it's not completely clear, so we refer to it as EV-depleted FBS rather than EV 3 fps. To allow quantitative tracing of these EVs in the mouse, I took a preparation of inflammatory activated mouse endothelial EVs, and then I transfected them with a non-mammalian microRNA, so MER39. It belongs to the worm, C. elegans, and it's not present in the mouse. So what you can then do is you can wash your transfected EVs and you can inject them into mice. You can then take tissues of interest and you can use reverse transcription PCR to look at the biodistributions of your EVs. And what we find is that they're delivered to the spleen in minutes and they remain detectable for up to 24 hours. This approach has some inherent limitations because we can't be certain that it's still EVs present at the later time points or whether it's just the transcript. But it does allow us to assess that endothelial cell-derived EVs are delivered to the spleen. So we're able to show that endothelial cell-derived EVs are enriched in the blood, they localize to the spleen, but do they exert any functional effects once they get there? To assess this, I took a preparation of inflammatory activated endothelial cell-derived EVs from the mouse cells and then injected them into mice. I then took the spleen and I looked at alterations in gene expression and then performed a protein chemokine array to see whether there are any differences. We find that the expression of CXCL1, CXCR4, interleukin-1 beta, interleukin-6 rapidly increase 2 hours post-injection, so a single injection of EVs, but not at other time points. And now this time point is important because it matches the time point of where we see neutrophil mobilization from the spleen in mice. The EVs are also rich in microRNA, and microRNAs can silence gene expression, so I wanted to know whether the delivery of these endothelial cell-derived EVs rich in the microRNA 126 could influence protein expression. I used the chemokine array here. We're looking at over 20 different chemokines, and we find significant reductions with 6 chemokines after a single EV injection, including for targets of 126, such as CXCL12, which is also called stromal-derived factor 1. My video is blocking the screen, bear with me, which is just up here. So when we normally think of chemokines, we think of their expression rising in response to inflammation. But CXCL12 is highly expressed in bone marrow and in the spleen. And what it does in these reserves is it retains neutrophils through very high expression. But microRNA 126 can downregulate CXCL12. And the lower expression of CXCL12 mobilizes neutrophils to peripheral blood. So informed by this, I wanted to know whether endothelial cell-derived EVs could mobilize neutrophils from the spleen to peripheral blood. So I injected otherwise healthy mice again with a single preparation of the inflammatory activated mouse endothelial EVs, harvested tissues of interest, and then looked at neutrophil populations using flow cytometry. There was a rapid and a substantial increase in the number of peripheral blood neutrophils. We get a simultaneous reduction in splenic-neutrophil number and a small effect from the bone marrow once it's normalized to the recipient mouse. So this supports our hypothesis that endothelial cell-derived EVs mediate neutrophil mobilization from the spleen. But what mediates the effect, and can we target this therapeutically? So it's well known that VCAM1 is expressed on activated endothelial cells, and the immune cells can interact with the VCAM1 through their receptor VLA4, which is expressed in neutrophils. We find VCAM1 enrichment on EVs at the time of injury, and we hypothesize that the inverse might be true, where plasma-liberated EVs enriched for VCAM1 could mediate neutrophil mobilization from the spleen following myocardial infarction. So to investigate this, I CRISPR-edited VCAM1 from some mouse endothelial cells to produce VCAM1 deficient EVs. Here's a Western blot, and what we normally see is that wild-type mouse cells, once they're exposed to pro-inflammatory TNF-alpha have a robust VCAM1 response, and now the knockouts have no response to the TNF-alpha. I've characterized the EVs from the knockout cells, but I'm not going to share that data with you. Instead, I'm going to focus on the functional experiments that I've undertaken. The knockout cells still produce EVs, and they're deficient for VCAM1. So we hypothesized that VCAM1 might be necessary for the endothelial cell EVs to localize to the spleen or to have functional effects, such as alterations in the gene expression or the mobilization of the neutrophils. To determine whether the VCAM1, which is enriched on the surface of the EV after MI and on the endothelial cell-derived EVs following inflammatory stimulation, is functional in determining the gene expression changes in the spleen, or whether it mediates splenic-neutrophil mobilization, I took wild-type mice. I injected them with the wild type with the VCAM1 knockout endothelial cell-derived EVs at the same concentration. And then 2 hours later, we take tissues of interest and we look at neutrophil populations using flow cytometry. And what we find here is that the VCAM1 knockout endothelial cell-derived EVs in the green fail to mobilize neutrophils to peripheral blood, thereby demonstrating a pathway where ischemic tissue liberates endothelial cell-derived EVs rich in microRNA 126, decorated in VCAM1 integrins. It allows them to localize to the spleen and to mediate chemokine regulation and mobilization of neutrophils. We wanted to know whether the gene expression changes that we had observed previously in the spleen were also dependent on the VCAM1. So following a similar experiment, I wanted to know whether the VCAM1 was necessary. So what we found was that VCAM1 knockout endothelial cells did not mediate changes in gene expression in the spleen, unlike the wild type cells, including for targets of microRNA 126, which are CXCL1, interleukin-1 beta, and IL-6. So in summary, acute myocardial infarction induces a peripheral neutrophil response that correlates with the extent of injury. Plasma EV number is elevated following myocardial infarction. It correlates with the extent of injury and the number of neutrophils found in peripheral blood. Plasma EVs are enriched for endothelial cell-associated microRNA 126 and the endothelial cell-associated integrin VCAM1 following myocardial infarction. These can localize to the spleen, they can down-regulate chemokines, induce alterations in gene expression, and induce splenic-neutrophil mobilization, which is dependent on VCAM1 on the EVs. Neutrophils are activated before they get to the site of injury, and they show enrichment for microRNA 126 targets, and targeting microRNA 126 with an antagomer lowers infarct size. So we've shown here a mechanism for neutrophil mobilization from the spleen that doesn't require systemic chemokine expression. And it might explain why plasma neutrophil numbers are elevated in patients before they get to the hospital and before elevations in systemic chemokines. Therapeutically targeting neutrophil transcriptomes to perturb their inflammatory effects before they enter the myocardium might be a novel opportunity to salvage the myocardium in the immediate hours from inflammatory damage. So thank you for your attention. Thank you to Opsami, to our funders, the British Heart Foundation, to the Centre for Research Excellence in Oxford, the many collaborators that I have who have enabled me to do this work. And if you have any comments or questions, I'd love to hear them.
Okay, can you hear me? Yeah, so very nice presentation. I learned about the extracellular vesicles, so it's amazing, yeah. And now we have some questions here, and the first question is why didn't you use the health patients as control? So use the NSTMI patients, so with the first questions anonymous. Okay.
Thank you for the question, anonymous attendee. The reason that we use NSTEMI patients is that the STEMI patients have underlying vascular disease of some kind. That's why they've experienced a myocardial infarction. And NSTEMI patients then are on appropriate control because they're roughly of the same age, they have underlying vascular disease, and then we're specifically looking at ischemic injury, which is what we're interested in, and not at some other pathology.
Okay. The next question, is there any evidence in the literature that exosomes are different to other EVs in terms of transcriptome? If so, could you please comment how does this affect your study? So this anonymous attendee also.
Short answer to that, I don't know. I'm just trying to digest the question. So are exosomes different from other EVs? Okay. So are apoptotic bodies, are microvesicles and exosomes, are they different following myocardial infarction? That detailed analysis, as far as I'm aware, hasn't been done. So a lot of the preparations are mixed populations. So you're either looking at apoptotic bodies, microvesicles, and exosomes. As far as I'm aware, no one has looked just specifically at comparing the exosomes to the microvesicles and over the evolution of the infarct.
But is it possible, Naveed, to do this? Is it technically?
It's technically challenging. So the markers that we use to derive either what we call exosomes or microvesicles aren't completely, they're a bit promiscuous. So some microvesicles might be carrying tetraspanins, and some of the so-called exosomes, they might be carrying some of the markers that we use for microvesicles. So it's quite difficult. So what you would need to do is, first of all, define how you're defining an exosome, and then to chase that up, potentially using a transcriptomic approach. But it's incredibly challenging from the biology point of view, and then from the isolation point of view.
Okay, so this nomenclature right now is sort of arbitrary, or do you think there is an actual sort of real biological reason for calling them different names?
So there is a real biological reason to calling them apoptotic bodies, microvesicles, and of exosomes. So exosomes come from an endosomal origin, and the microvesicles are budding or blebbing from the membrane. And so what you can do in culture is you can determine this. So you can use various trackers, you can stain up your lysosomes, and you can show that they're either producing your exosomes or they're not. When you move into complex organisms, such as the mouse or the human, and then complex biological fluids, it's pretty difficult to determine where they may have come from.
Thank you.
Okay. So here I have the next question is, you mentioned in the beginning that chemokine concentration levels were too low to account for this recruitment of neutrophils post-myocardial infarction. If EVs are the recruitment signal instead, what is the concentration of EVs in the heart or spleen in these patients? The first question. And in the sequence, the same person, anonymous attending, asked how do these EVs manage to find and get taken up by the appropriate neutrophil subpopulations versus all of other circulating or resident cell types in the body?
That's two great questions, so thank you.
Yeah.
So the first question, so the EVs are the recruitment signal. What is the concentration of the EVs in the heart or in the spleen? So what we haven't done, but other investigators have done, is that they've taken the heart following myocardial infarction, and they do a small digestion to look at the number of EVs that are still trapped within the myocardium. And what they find is that endothelial cell-derived EVs are the principal source. What, as far as I'm aware, that no one has done is to look at a human spleen and to say, can I directly find endothelial cell-derived EVs, principally because we can't take a spleen from a human patient, especially if they've just had a myocardial infarction. And then your second question, I think your question came maybe slightly before I shared the data. So what allows the EVs to be taken up by the appropriate neutrophil population in the spleen. So we believe it's the VCAM1. So the VCAM1 on the endothelial cell-derived EVs is important in signaling to the neutrophils to mobilize. What the absolute mechanism of that signaling is at the moment, we don't know. But what we do know is that the VCAM1 doesn't seem to be important for localization to the spleen, but it's important for exerting the effects.
Sorry, Naveed, do you mind if I jump in again with this?
Of course.
Because I've noticed in your data that actually your VCAM levels are much higher in humans than there are in mice. Does that mean that the neutrophil mobilization is much stronger or greater?
What we didn't control for in the data that I've shown you, obviously human is much larger than the mouse. We're able to get a lot more blood from our patients. So what we haven't done is fully started the same amount of starting material there. That's something that we can look into.
I thought it was normalized per milligram of total protein.
Per each assay, but not between the species.
Okay.
It's because I showed them side by side.
Okay.
Okay. So the next question is, could you elaborate on the apoptotic bodies? Are they included in your EV's population? And if so, how do you characterize them? Thank you.
So as far as I'm aware, the apoptotic bodies are not present in our EV preparations. So as we take our blood from our patients, we initially produce a platelet-poor plasma. So we initially produce the plasma, then we take the plasma and we centrifuge it again to remove the platelets, and then the plasma is stored. When we're ready to isolate the EVs, we then give it a relatively hard spin, around 5,000 G, which should remove any remaining debris. And those types of spins are sufficient to remove the apoptotic bodies. If I then stain our EV preparations for stuff that might be reminiscent of a dead or a dying cell, such as mitochondria, or for histones, we don't detect those in our plasma EVs. And so I'm with some certainty, I think we can say that our EV preparations are more microvesicle and exosomal small EV rather than apoptotic bodies.
Okay, so the next question is from Irving Zucker. Do plasma EVs post myocardial infarction show cardiac myocyte markers or microRNAs that are specific to the heart? This is one of my questions also. And the second question from Irving is, can the spike in the IVs be from extracardiac sources and that may show impaired blood flow and hyperfusion following myocardial infarction?
Okay, thank you for your question.
Irv is the editor of AJP Heart, so you need a good answer now.
Okay.
I'm trying to read and come up with the answer at the same time. So there are a number of published studies where they've looked at cardiac-specific markers. So microRNA1 is one of those, which is regarded as a cardiac microRNA marker. It was present on our array, but what we're not looking at in the array is just the absolute change, but the weighted change. So we feed the data into a regression model, and we say, okay, the patients are of different age, different BMI, they had varying sizes of infarct, their troponin levels changed not potentially consistently across the entire cohort. But if we feed all of this into the regression model, what are the microRNAs that then stand out? And in our list of 12 microRNAs, there's no, MYR1 doesn't come out in that. And then, so I'm just reading your second part of your question.
Okay, yeah. The next question is from Sina Handpour Lex Meshari. I don't know if that's correct, sorry. Interesting talk. Are there any changes in neutrophil or plasma EV levels with age?
Sorry, but Naveed, have you responded to the second part of that?
Yes, yes. So I was just reading it. So once the myocardium, that's okay. Once the myocardium is injured, we know that you get activation of distal vascular beds. So following myocardial infarction, you can show VCAM1 expression and immune cell recruitment to the kidneys. So are all of the EVs that are present in the plasma coming directly from the myocardium? We can't say that completely because we don't have organ-specific endothelial cell extracellular vesicle markers. We don't even have cardiac cell type EV-specific markers, let alone go into a different organ. So that's difficult to determine. I think it would be interesting to look at patients who show good perfusion or poor reperfusion and whether we get differences in the EV response or the EV microRNA profiles. We could possibly do that with the number of patients that we have at the moment, but there's only around 15 patients in the study that I've shown you today, so it would really be dwindling the numbers.
Okay. So the next question is from Emma Robinson. And she said, thank a lot. A couple of questions. So here we have a lot of questions. I missed the first slide or two, but from where did you extract the blood? So it's the first question, okay? Normally, blood would be extracted from superficial veins, close the skin in the humans. This would not be a source of blood. It's between the heart and the spleen, and likely, in fact, depleted of extracellular vesicles that are produced and secreted by the heart, as they have been taken up by peripheral tissues. Did you simply treat neutrophils in vitro with patients with patient EVs versus healthy, no acute myocardial infarction, and EVs, and observe phenotype and gene expression changes versus mRNA mimic transfection?
Okay, thank you for your detailed questions, Emma. We missed out one question just slightly above, so I'm just going to answer that first of all.
The first question.
Are there changes in neutrophils or plasma EVs with age? The answer is yes. So there's been a number of population-based studies that show changes in immune cells with age and with the concentration of EVs. The concentration of EVs was a scientific reports paper. I can't remember the author's name though. So just coming on to yours, Emma, sorry, I'm just gonna have a quick read again.
Okay, the next part of this question, did you simply treat the neutrophils in vitro with patients' AVs versus healthy?
No, Marcella.
Yes, sorry, I'm
so
being
that's
slow.
one question before. I'm
reading.
Sorry, yes.
So the blood samples that we take are from the catheter that is put into the femoral artery. So as we take our blood samples before they receive clinical therapy. So that's where we get them from. The follow-up blood samples do come from a peripheral vein, because we're not going to insert another catheter into them just for a follow-up blood sample. So I completely take on board to what you're saying, that we may have lost some of the EVs because of clearance from the circulation, but those are the samples that we currently have at the moment. What we also have stored are coronary sinus blood samples, so blood from the coronary arteries themselves. So we have that before they're stented and after they're stented. And we're hoping to look at these samples. These are very, very small, minute blood samples. So we don't have as much material to use as we do with peripheral samples, but it would be interesting to see, are we losing some of the signatures that we're detecting in peripheral blood that we can detect in the coronary sinus? But ideally, what we would like to do, or what we envisage, is that these extracellular vesicle signatures, whether it's the protein or whether it's the microRNAs, could be used for some diagnostic therapy or to guide treatment of patients. So peripheral blood would be the initial preference. but we do have access to other samples. And sorry, just to read your next part. So if I treat neutrophils, whether they're mouse or human, with EVs, we are able to induce alterations in gene expression. That's not data that I've shared today because there's a lot of it, in all honesty, but we do observe changes, yes.
Okay, so that's okay now, yeah. So the next question is from Sarah Braba Mendel. Talks was very interesting. Dr. Naveed, my question is, do both HSR microRNA 126.3p and 126.5p were functional at the same time? The first question. And the second is, have you tried an experiment using to understand which one was functional?
We haven't done experiments to look at whether 126.3p and 5p are functional at the same time, but I can say that 126.3p is more abundant. In the endothelial cell derived EV, so from culture following inflammatory stimulation, 126.3p is more significantly enriched than the 5p. Certainly with the experiments that I've done.
Okay. Here the next question is from Kaloyan Takov. So thank you, Naveed, great talk, and a few questions. So you can do separate, okay? So the first one, have you checked the yield of your EVs isolation approaches by measuring the residual EVs and microRNA 126 in the supernatant EV depleted fractions? Previous studies showed that UC pellet and EV-rich second fractions contain only a small proportion of total microRNA 126 detected in plasma.
Okay, just to answer, thank you for your question, Takov. I recognize your name, and based on your questions, I assume you work with extracellular vesicles. So the observation that we've made that you get enrichment for 126, the data I've shared with you today was ultracentrifugation with washing. I've also done size exclusion chromatography combined with ultracentrifugation to give us a cleaner prep, and we still detect it as significantly enriched in patients following myocardial infarction. So your second question?
Second question was, lipoproteins are co-isolated UC and SEC from plasma, and have you been shown to carry microRNAs? Have you checked the lipoprotein levels and potential interference?
So I completely agree. So ultracentrifugation and size exclusion isolate or co-isolate lipoproteins, which can also carry microRNAs. We are in the process of moving on to trying to use immunoaffinity capture to get around this. So I haven't specifically looked at lipoproteins within the EV fractions, but that's a great suggestion.
Okay. And the last question from this, from the Kaloyan, have you seen any phasic response for extracellular vesicles and neutrophils levels in blood similar to different immune cells after acute myocardial infarction? Example, any early have or increased cardiac extracellular vesicles in the blood followed by increased neutrophil number later?
Yeah, so this is what I think is happening. So based on our investigations, I think that there might be an early release of endothelial cell-derived EVs which mobilize the neutrophils. So in the data that I showed you, if we perform experimental myocardial infarction in the mouse, we find that the neutrophils mobilize from the spleen. But what we don't find are alterations to the monocytes. We know that monocytes do mobilize from the spleen. This was shown a number of years ago, but it happens a lot later on. So how do the signals change? And so EVs can mobilize both monocytes and neutrophils. So if you inject endothelial cell-derived EVs, both of them will leave at the same time. So it is possible that in vivo you get certain phases of EVs being released by the injured myocardium. What we would like to do is, because we're able to get serial samples from our patient, so before they have clinical therapy and then they're in hospital for up to 2 days, we have serial blood samples stored for them, is to try and look at these. Are there smaller differences that we're not detecting at the moment because of our group size or the techniques that we've used? But it's a great question. It's something that we'd like to look into.
Okay. The next is not a question, only to thank you for the great talk from Jan Jackson. The next is from Sam Datz. Nice work. Thank you for sharing. Just wondering if you just inject VCAM1 instead for EC extracellular vesicles, can you stimulate neutrophil release from spleen?
Thank you for your question, Sam. So we haven't done this. We haven't injected just VCAM1 as a recombinant protein. But what I do know is that VCAM1 is chemoattractant. So if you do something like a boil-in chamber, you can make immune cells migrate towards VCAM1.
Okay. So the next is from Kumar's Mobile. Nice talk. Do you mean neutrophils binds to VCAM to induce the inflammation?
Thank you for your question. So we know that the VCAM1 is necessary to get the changes in the gene expression and to get the changes and to induce the neutrophil mobilization from the spleen. What we're uncertain of at this moment in time is what is the absolute interaction. Is the VCAM1 interacting with the neutrophil? You could possibly cause calcium buffering, which can induce motility in immune cells, or is it affecting some way that they're tethering? We're uncertain at the moment.
Okay, so Derwin Zucker again, good talk. We'll send your papers to review. Call for papers on exosomes going on now. Just a comment, I know.
There you go. Any papers, send them to AJP Heart.
Thank you very much.
Okay. The next question is from anonymous attendee. Thank you for a great talk, Naveed. Would you like to comment on how much of the contribution to reduce infarct size is due to the direct effect of exosomes on neutrophil recruitment compared to exosomes' effect on all immune cells and the resulting crosstalk?
Sorry, I'm just trying to digest your question. It's a very large question.
Yes.
So we know that the EVs are mobilizing the neutrophils. So an interesting follow-up question is, do the neutrophils, once they're mobilized, are they more prone to undergo recruitment to the injured heart? We don't know that at the moment. One of the other things that we don't know at the moment is whether the injection of the EVs is going to make the infarct better or worse. So that's another outstanding experiment for us at the moment. And so the second part of your question, we don't know at the moment.
Okay. So here I have a question from Heinrich Tagmeyer. I don't know if that's correct, sorry. Beautiful. Thank you, Naveed. Quite question. What is the role of the bone marrow? The reason for my question is bone marrow metabolic activity can be assessed with FDG.
So again, just to say, legend of cardiovascular.
Yes.
I'm saying Maya. Hello, Henrik.
Thank you for your question. So what we're not doing is we're not disregarding the bone marrow and its role in contribution of neutrophils to myocardial infarction. This has been investigated for a couple of decades. What we're trying to determine is, why do neutrophil numbers rise so quickly before you get these elevations in these chemokines? And before or as the patients come into the hospital, is there a potentially another source of neutrophils that is mobilized early, which can then start priming the myocardium, start releasing more chemokines to signal to the distal bone marrow? And that's the axis that we're working on. So it's not to say that the bone marrow isn't an important source, because it is. And what we also don't know is whether the neutrophils that are in the spleen, did they come from the bone marrow, which we assume they do because we don't have any evidence to suggest that the spleen can generate neutrophils.
Okay, and here we have the last question for a while is thanks from Shanghai Tian, I don't know. Thank you. Do you think microRNA 126-3P and 5P share the same targets of different targets? Which strains will be more important in acute myocardial infarction?
Thank you for your question. So it's a great question. So in the in silico bioinformatics analysis that we've done, I have combined 3P and 5P targets together and call them microRNA 126 targets to look at the combined list. But we could look at those as separate lists and then look at the overlap between them. But I haven't done that yet.
Okay, so here we have one more question from Rio Juni. VCAN1 is endothelial cell activation marker, normally expressed on the surface of the endothelium upon pro-inflammatory stimulation, for example. As EVs contain a lot of cargos, what do you think about the other chemokines in the EVs that may attract neutrophils?
Thank you for your question, Rio. So I know that there are a couple of studies where they use endothelial cells activated with various agonists, and they show that there's chemokines present within these exosomal fractions. So yes, we know that the EVs can contain chemokines. When does that happen though in myocardial infarction? So we're saying that you potentially get an early release of extracellular vesicles from the endothelium as it's undergoing activation. So the assumption would be that endothelial cells might have pre-packaged EVs sitting inside the cell, packaged up with chemokines, or do they package in the chemokines later on? And so those are two things that we just don't know at the moment.
That's okay. So we have one more question right now. So Jung-Yu Sun, will the protective effect of ischemia preconditioning able to be blocked by inhibiting exosome pathway? Why is the most effective way to inhibit the exosome? This is a good question.
Okay, so we haven't looked at preconditioning, but there are groups who look at this. Exosome inhibition is difficult. So if you look at the literature at the moment, the number one inhibitor that is used is GW4869, neutral sphingomyelinase inhibitor. But if you inject this inhibitor, which people do, into animals, you're going to affect every pathway that's dependent on ceramides. So that's not an avenue that we've gone down at the moment because we think it's a very messy approach. So even using it in culture, you have to figure out the dose that you're going to use, and not all cells are using the same pathway to produce EVs under basal conditions or under stimulated conditions. And that, again, is then cell type specific as well.
Okay, I think there is no more questions. Yeah. So I'd like to thank you for this very amazing presentation. And now I'm very excited to study a lot more extracellular vesicles. So there's the future, yeah. And I have a doubt about the preparation of this isolation of these extracellular vesicles. So do you have contamination? Yeah, I think this is a little part, but you have the contamination. And how can I exclude these interferences in our results? So it's very difficult to answer this question, but I want to know if it's more simple to isolate the extracellular vesicle from the tissue or from the body, for example.
So I haven't looked at whole tissue preparations. So
Okay.
there are groups that take the injured heart, they'll do a bit of digestion or cutting of the heart, and then look at trapped EVs. We haven't done this. So I've principally looked at cell culture supernatants, urine, or looked at plasma. And so we know that there are inherent limitations. with the methods that we use. So the most popular is ultracentrifugation. You can overcome some of the limitations. So the way that the field has been moving is ultracentrifugation, most popular method, very easily accessible. But what you might do is you might co-isolate some lipoproteins and some soluble proteins. So one way around that is to use size exclusion chromatography. And then you can combine that with ultracentrifugation. So you can elute off soluble proteins, but what you can't get rid of are your lipoproteins in your plasma, for example. And so you have got rid of one potential contaminant, but you can't get rid of the contaminant completely. But what I should say is it depends on what you want to do. So if your principal purpose is to say, I want to know what EVs are there, what is the number, what is the characterization of those, you can use very advanced flow cytometry techniques and determine that. But what you're not going to be able to do then is to take those EVs and then put them into a functional assay. So they're going to be either collecting in the effluent from your flow cytometer, or they're now covered in antibodies, or they're covered in magnetic binding particles. So even with our approach where we can select the VCAM1, we then do the analysis as a delta, as a subtraction. What we can't do is you can't get them back off the bead. So that is currently missing within the EV field at the moment. How can we select very pure subpopulations and then functionally determine what their effects are versus the rest of the population, or the swamp, if you want to call it that. So.
But, you know, Naveed, I think you're not alone in this. You know, even if you look at cell types, you know, a simple cardiac fibroblast is actually something difficult to identify compared to other cell types. You know, search for those markers is not easy, even with established cell types, let alone with new field like, you know, vesicles. So I think this probably will be an ongoing search for the methodology.
Certainly. So even if you use some of the more robust methods for getting pure preparations are these immunoaffinity capture, but you can't, if you're looking at a plasma, you're using the same EV marker that's expressed by skeletal muscle, cardiac muscle, endothelial cell, hepatocyte. And because our knowledge of where the EVs are generated is the same across all cell types. So we're using the same markers across all of these cell types. Up until we start getting tissue-specific or cell-specific signatures out or subpopulations, we won't be able to really draw on those kind of diagnostic potential there.
Sure. But I guess size exclusion chromatography, you know, is helpful there. You know it's smaller than a cell, but still trying to get that biomarker for that particular subtype of the EV is going to be a tricky one, right? I suspect. But.
Okay. So thank you very much. Denver, do you like to say something?
No, I'd like to thank Naveed, first of all, for a great presentation. I think very logical, very detailed. I mean, I've certainly learned a lot. And of course, you know, thank you, Marcella, for chairing and making this a truly international affair. And of course, I'd like to thank the attendees for sticking with us, and see you all again on Thursday, hopefully.
Okay.
Thank you, David. Thank you, Marcella.
Thank you.
Thank you. Bye, everyone.
Bye.